Picture yourself on Mars. The environment, even the atmosphere, is totally different. Do you behave the same way you do here? Probably not. But this is essentially what we do to cells when we use them to test new drugs like cancer treatments in environments like petri dishes before they're tested in animals or people. And just like for us, the environment around the cells can impact their behavior and their response to the drug. So it's no wonder that almost 95% of cancer drugs fail during clinical trials. The goal of my thesis is to make new devices for testing cancer treatments that bridge the gap between petri dishes and clinical trials by better mimicking the natural environment of the cells in the body. Now, one crucial aspect of the cell's environment is oxygen, and oxygen is especially important in cancer research. The blood vessels in tumors can be abnormal, which causes parts of the tumor to have very low oxygen. The blood vessels can also close and then open again, sometimes a few times per hour, which exposes the cells to cycles of low and then normal oxygen. Cells that are exposed to these low and cycling oxygen levels can be more aggressive and less treatable. So incorporating these conditions into the early stages of testing could help us to better predict the response in the body. Now, the problem with incorporating these conditions into a petri dish is that the gases that are used to control the oxygen levels are too far away from the cells. Just like it takes you longer to run 10 kilometers than 1, it takes the oxygen hours to move millimeters through a petri dish, but only seconds to move a fraction of a millimeter in the body. So those switching times of a few cycles per hour in tumors can't be mimicked in a dish because the oxygen levels can't change fast enough. So to solve this problem, I use engineering to shrink everything down. I use tiny channels with walls that are made of a material that oxygen can pass through. The cells are grown in one fluid-filled chamber in the device, and control gases flow through very close but separate channels. Oxygen from these gas channels can pass through the walls, moving less than half a millimeter to control the oxygen around the cells. Over the first few years of my PhD, I've made this device and verified that it can create the kinds of low and cycling oxygen levels that are found in tumors, enabling studies that couldn't be done in environments like petri dishes because of those large size scales. Over the next year, I'll be using the device to understand the effect of these different oxygen profiles on cancer cell behavior and response to treatment. My thesis is a step towards recreating the actual environment of tumor cells in the body. And the better we can do that, the better chance we have of getting early stage test results that predict the outcome in patients, which would speed up the development of new, effective cancer treatments. Thank you.